We have now talked about how we create our electrical signal, and now we will talk about how we transmit the electrical signal. Signal transmission will occur at the synapse. A synapse is a functional junction between one neuron and another neuron, or between a neuron and an effector, such as a muscle or a gland, if you recall from our first video for this chapter. There are two types of synapses, electrical and chemical. Electrical synapses occur via gap junctions connecting the cells and allowing the transfer of information to synchronize the activity of a group of cells. The ionic current spreads directly from one cell to another cell through the gap junctions. Electrical synapses allow for faster communication and will synchronize the activity of a group of neurons or muscle fibers, such as our cardiac muscle in the heart. Chemical synapses, there's a one-way information transfer from presynaptic neurons to a postsynaptic neuron. So this will occur in one-way transfer, as a one-way transfer only. Here we show the chemical synapse. We are showing the nerve impulse here in the presynaptic neuron. The calcium is the chemical, and this calcium is going to be released from as the neurotransmitter into the area called the synaptic cleft, and it will reach the postsynaptic neuron, creating a postsynaptic potential and a nerve impulse. This should look very similar to what we had at the neuromuscular junction. However, here we are showing you nerve and nerve. So presynaptic and postsynaptic neurons. So two neurons, but very similar to the synapse point we had at a neuromuscular junction. Just here we have a presynaptic neuron and a postsynaptic neuron. So a little different junction. Another great 3D physiology animation for you here. At this synapsis, we will have one of two postsynaptic potentials created, either an excitatory postsynaptic potential or an inhibitory postsynaptic potential. This postsynaptic neuron is an integrator, meaning that it receives and integrates signals before creating a response or an effect. The effect that it creates will then either be excitatory or inhibitory. If the excitatory effect is greater than the inhibitory effect, but less than threshold level of stimulation, our result is a sub-threshold excitatory postsynaptic potential, making it easier for us to generate a nerve impulse. If the excitatory effect is greater than the inhibitory effect, and we reach or surpass threshold, then our result is a threshold or a suprathreshold excitatory postsynaptic potential, and a nerve impulse is created. However, if the inhibitory effect is greater than the excitatory effect, the membrane will hyperpolarize, creating an inhibitory postsynaptic potential with failure to produce a nerve impulse. A postsynaptic potential neuron can receive many signals at once. Neurotransmitter receptors at chemical synapses can cause either, as we said, an excitatory or inhibitory graded potential. Neurotransmitter receptors have two structures: ionotropic receptors and metabolic receptors. Ionotropic receptors will be ion channels that directly bind the neurotransmitter, whereas metabolic receptors are proteins, specifically G proteins, that bind to the neurotransmitter but are not the ion channel. Instead, these binding proteins initiate ion flow through another protein. An excitatory neurotransmitter is one that can depolarize or make less negative the postsynaptic neuron's membrane, bringing the membrane potential closer to threshold. An inhibitory neurotransmitter hyperpolarizes the membrane of the postsynaptic neuron, making the inside more negative and generation of a nerve impulse more difficult. A hyperpolarizing postsynaptic potential is inhibitory and therefore termed an inhibitory postsynaptic potential, or IPSP. So here we see our ionotropic and our metabotropic receptors. We see our resting membrane potential with our ionotropic here of acetylcholine, causing us to have an excitatory postsynaptic potential. Here we see the binding of GABA in the ionotropic receptor, leading to this inhibitory postsynaptic potential. And then finally we see our metabotropic acetylcholine receptor, causing us to also have an inhibitory postsynaptic potential. So with this being said, you see here that acetylcholine was excitatory at one location, and then it was inhibitory at another location. So the same chemical can cause either an excitatory or inhibitory potential dependent upon the location. A neurotransmitter must be removed from the synaptic cleft to cease the impulse, and this will happen by diffusion, enzymatic degradation, or by the neurotransmitter being taken into cells, such as the neurons and the glia. Next is summation. Remember we said that summation was taking more than one synapse at the same time and creating a combined effect that might generate a nerve impulse. So if we have several presynaptic end bulbs, we're showing 5 here, releasing their neurotransmitter at about the same time, this combined effect may generate a nerve impulse due to summation. Summation can then be spatial or temporal. Here we show that spatial summation will have several nerve impulses, and then eventually we will be able to reach threshold and create our nerve impulse over time. Whereas temporal summation, the impulses all pretty much occur roughly at the same time, but they're just a little stronger each time, and so we get finally to threshold and we'll reach our action potential. You have a summary of the neuronal structure and the basic functions as well. The neurotransmitters that we've spoken of can both be excitatory and inhibitory and will be present in the central nervous system and the peripheral nervous system. As we saw with acetylcholine, the same neurotransmitter may be excitatory in some locations and inhibitory in others. Many of our neurotransmitters are also hormones released by endocrine glands, whereas some brain neurotransmitters are released by neurosecretory cells. There are small molecule neurotransmitters and neuropeptides. The important small molecule neurotransmitters include acetylcholine, the amino acids glutamate, aspartate, gamma aminobutyric acid or GABA, and glycine. We also have biogenic amines, norepinephrine, epinephrine, and dopamine. The important neuropeptides include endorphins, hypothalamic releasing and inhibiting hormones, angiotensin II, and cholecystokinin. This just shows the chemical structure of the neurotransmitters and breaks them into the small molecule neurotransmitter groups and shows you the neuropeptides. This table includes some of the neuropeptides and their descriptions. Neurotransmitters can be modified by stimulating or inhibiting the synthesis of the neurotransmitter. So if we create more or create less of the actual neurotransmitter, that will modify it. If we block or enhance the release of the neurotransmitter. Also, if we stimulate or inhibit the removal of the neurotransmitter. And finally, by blocking or activating the receptor site, so where the neurotransmitter would bind. Any one or combination of these things can modify our neurotransmitters. Regeneration and repair of nervous tissue. Throughout our life, the nervous system will exhibit what we call plasticity. And this is the capability for change with learning based on experience. Even though we have this plasticity, neurons have a limited capacity to be able to repair or replicate themselves. That repair and replication is going to be what we call regeneration. In the central nervous system, there is little or no repair of damage to neurons due to inhibitory influences from the neuroglia, particularly the oligodendrocytes. Also due to the absence of growth-stimulating cues that were once present during our fetal development, and there is rapid formation of scar tissue that occurs. Now, in the peripheral nervous system, damage to dendrites and the myelinated axons may be repaired if the cell body remains intact, if the Schwann cells are active and functional, and if scar tissue formation does not occur too rapidly. If those things are the case, then we have 3 steps involved in the repair process: chromatolysis, Wallerian degeneration, and formation of a regeneration tube. When there is damage to an axon, usually there will be changes called chromatolysis, which occur in the cell body of the affected cell. This causes swelling of the cell body and peaks between 10 to 20 days after injury. By the 3rd to 5th day, degeneration of the distal portion of the neuronal processes and the myelin sheath, Wallerian degeneration, will occur. Afterwards, macrophages will begin to phagocytize the remains and will have retrograde degeneration of the proximal portion of the fiber, extending only to the first neurofibril node. Regeneration follows chromatolysis. We'll have synthesis of RNA and protein accelerates that will favor rebuilding of the axon, which will often take several months. Disorders that affect the nervous system include multiple sclerosis, epilepsy, excitotoxicity, and depression. Multiple sclerosis is an autoimmune disease that results in progressive destruction of myelin sheaths and neurons in the central nervous system. These myelin sheaths in the central nervous system deteriorate to what we call scleroses, which are hardened scars or plaques in multiple regions. Over time, more and more myelin sheaths sclerose, making this a progressive, debilitating disease. Epilepsy is the second most common neurological disorder after stroke. Epilepsy affects 1% of the population. It is characterized by short, recurrent, periodic attacks of motor, sensory, or psychological malfunction called epileptic seizures. So these epileptic seizures are short, recurrent, periodic attacks of motor, sensory, or psychological malfunction. These seizures are often initiated by abnormal synchronous electrical discharges from millions of neurons in the brain, perhaps resulting from abnormal reverberating circuits. There are many causes of epilepsy, but the most common cause is brain damage at birth. Other causes include metabolic disturbances, infections, toxins, vascular disturbances, head injuries, brain tumors, and brain abscesses. However, most epileptic seizures are idiopathic, meaning that they have no demonstrable cause. Epileptic seizures can be eliminated or alleviated by drugs that depress neuronal excitability. Speaking of excitability, excitotoxicity causes high levels of the neurotransmitter glutamate to cause destruction of the neurons due to oxygen deprivation. And our last disorder here is depression. It is another disorder of the nervous system. People who are depressed may feel sad and helpless. There are many types of depression, and we have listed some here. Major depression, in which symptoms last more than 2 weeks. Dysthymia, in which people alternate between depression and periods of normal. Bipolar disorder, which is a manic depressive illness with periods of extreme elation, also known as mania. Lastly discussed here is seasonal affective disorder, or SAD, in which depression occurs during winter months. This will conclude Chapter 12, Nervous Tissue.